Okay, I think we are good to go here. Welcome, welcome, welcome back. We are in lab 9. Oh, not 9. uh, wow. So we have 2 labs left. It's just this lab and the last lab. And so thanks for joining me once again, and let's get to it. So the last class we talked about the vascular plants, the spore plants of the vascular plants, ferns, lycopods, and horsetails. And then we talked about the alternation of generations in those plants. Today we're going to talk about the gymnosperms, which we have encountered a little bit already during the first lab. But in particular, we're going to talk about cycads, which we haven't encountered yet, ginkgo, and conifers, which we have encountered, and the alternation of generations as it pertains to those plants. So let's do it. As we know, we are well up into the vascular plants. We covered those spore-bearing vascular plants last time. Now we are up into the seed plants. We've already talked a lot about the seed and the seed germination lab, but we are now going to talk a little bit more specifically about these plants. And just a reminder, there are 2 types of seed plants. There are the gymnosperms, which we'll talk about today, and the angiosperms, which we'll talk about in lab 10. And a little reminder here that reason why we call gymnosperms is because the seeds are not in a container. The seeds are exposed or naked, not enclosed in a vessel like an ovary. And so that means that you can see the seeds with the naked eye. And before it was a seed, it was an ovule. And so you can see the ovules with the naked eye as well, because they're not in a container. Whereas the angiosperms, the ovules are inside of an ovary. And once the ovules are fertilized, they become seeds, and then that will be inside of a fruit. Talk about them more next time. We've seen this schematic before, but this is just showing you the differences. The blue thing on the left is the ovule of the gymnosperm, and the red thing on the right is the ovary of the angiosperm with the ovule inside, protected. So this is what today we're going to talk about gymnosperm cycads first. So this is just a quick picture of cycads, then ginkgo, and then conifers. So let's talk about the cycads first. So cycads are in the phylum Cycadophyta, right? And the way you pronounce it is there at the bottom, Cycadophyta. I don't know if you could hear that. Somebody's backing down the block. And the reason it's called Cycadophyta, it comes from the Greek kaikos. I think that's how you say that. Koikos. And that means palm. And so whoever named it thought cycads looked like a palm. But it's not a palm. It's not a palm. Cycads in evolutionary time are millions of years older than palms. So it is not a palm, but it does, fair point, does look like a palm. And there are only about 300 species. You don't need to know the number. I'm just showing you that it's not that many. And they're actually experiencing decline as well because of these factors. So they reproduce very, very slowly. They have seed predation, so things eat the seeds. And I guess that makes it so the seeds are somehow damaged by being eaten. Habitat is being destroyed where they live, and so they can't live there anymore. And in some cases, governments remove them because the seeds can poison livestock. So you don't need to know these millions of years ago, but I'm just trying to show you how old they are. So the cycads arose during the Permian, and there was a lot of diversification during the Jurassic period. Jurassic. So that means that cycads are plants the dinosaurs ate. So the dinosaurs ate cycads. This is something we always, it's always like, what? Yeah, but this is plants that dinosaurs ate. And I think these pictures are super funny because they look so old. But yeah, plants the dinosaurs ate. Pretty cool. So the lab says that they are found worldwide. Well, they were once found worldwide, but now they're only growing in the tropical and subtropical regions, particularly Mexico, Central and Southern America, Southern Africa, parts of Asia, Australia, and South Pacific. And there is a native species. So I don't know if we've talked about this before, but the concept of native versus non-native is kind of it's a human construct because it's based on a point in time. But it's still a language that we have to deal with. And so let's put it this way. And native and non-native means different things depending on where you are in the world. So in the United States, unsurprisingly, if something was there prior to the 1700s, it's considered native. And if it was there after the 1700s, it's considered or found later not. So it's a human construct. I don't particularly care for the terms, but there is a species, let's put it this way, there's a species in Florida that has been there at least since the 1700s. That's my opinion on that. Let's see. Okay, so cycads have two types of habits. There's a columnar habit and then a bulbous habit. Columnar just means like a column. Bulbous just means round and short. So the one on the left is columnar, the one on the right is bulbous. The roots are a taproot system, which we learned about during the root lab. And the leaves tend to be compound leaves, and they have the same parts that we've already covered: blade, lamina, rachis, and petiole. And like I said, they tend to be compound leaves. They are megaphylls, like the ferns and the horsetails that we've already covered. Yeah. And what else? Oh yeah, so like there tend to be 1 times pinnately compound. That tends to be their leaf shape. Here it is again. I think we saw this before in the leaf lab. Just a reminder, the whole thing there, that whole thing there that the person's holding is a leaf. And the little leaflets, right, are what we call pinna. All right. So there's going to be some terms here you don't need to know, but they're on the slide. So I'm just showing you. So I will tell you what you need to know, right? If there's a term on any of these slides that are coming, you don't need to know them unless I mention it, okay? So the ovules, so we're in the seed plants, which means we have ovules now. We didn't have ovules in the ferns. We didn't have ovules in the lycopods. We didn't have ovules in the horsetails, right? We have ovules now. We have ovules now. And inside of that ovule is the Megasporangium. So we talked last time about how in Selaginella, for example, we had 2 types of spores, right? We had the microspore and the megaspore. Microspore carried the male gametophyte inside. Megaspore carried the female gametophyte inside. And so we can talk about the sporangia that they come from with those same prefixes, right? So Megasporangium is going to make megaspores. And that is located inside of the ovule. Likewise, Microsporangia are going to make microspores. So this is a close-up of we'll talk about it in a second. And this thing that I've circled that kind of looks like a little yellow Pac-Man, that is one microsporangium. Okay. Let's see. Yeah, so the ovules, which have the Megasporangium inside, and the microsporangia, in the case of cycads, are attached to special leaves. We've already a little bit, I think, covered this in this lycopod section of the last lab. Megasporophyll is going to have the ovule on it. Microsporophyll is going to have the microspores on it. The megasporophylls and cycads are sometimes arranged in astrobolus, and other times they are not arranged in astrobolus. It'll depend on the species. Whereas the microsporophylls, right, the leaves that bear the microsporangia, are always going to be arranged in astrobolus. Always. Okay. Remember, we've already come into contact with astrobolus before, right? Selaginella had strobili. The lycopod sometimes had, the lycopodium sometimes had strobili. And there was even astrobolus or strobili in the horsetails, right? So we've already come in contact with that concept. Just a reminder, it's a stem, and it has special leaves on it, and the special leaves have the sporangia attached to them. So I'll just draw here, right? Here is a stem. These are 2 different strobili here. Each of these kind of weird round things is actually a special leaf. I know it doesn't look like what you would think a leaf looks like, but they are technically special leaves. And on each of those special leaves, right here, there, here, all these guys down here are red, those special leaves will have on them the sporangia. So that's, remember, strobilus is just a way to get more sporangia in a smaller location. So the leaves have been modified over evolutionary time to be really small and round because they're not doing photosynthesis. Their job is only to hold a sporangia. There's a lot of variability in color and shape and size in the strobili of cycads, which is very, very beautiful. Somebody could spend their whole life probably studying the diversity in cycad strobili. So I'm not going to do that, but somebody could do that, and they're very pretty to look at. So cycads are strictly dioecious. Remember those terms from the very first lab: monoecious, one house; dioecious, two houses, the house being a plant. So if you're monoecious, you have the male and female sex organs in one plant. If you are dioecious, in the case of which cycads happen to be, you will have male and female sex organs in two separate plants. The female plants only bear the female sporophylls, right, megasporophylls. And the female plants have, depending on the species, the female plants have their megasporophylls arranged either in a strobilus or in what we call a crown of fertile leaves. And we'll see what that looks like here. So on the right is an example of a female plant whose megasporophylls are arranged in a strobilus. On the left is a female plant whose megasporophylls are arranged in a crown of fertile leaves. We'll look at what the leaves look like on the following. Sometimes the book or other sources might call cycad strobili cones. They are not cones. They are not cones. I can write a long thesis about how they're not cones. It has to do with what they are, what they consist of. Basically, strobilus is a stem and leaves. A cone is a branch system. So we'll talk about cones later in this class, but strobili are what cycads have. They do not have cones. And like I said, this is, so here you go. This is a female, this is from a female plant. This is a species whose megasporophylls are arranged in strobili. And you can see here those little kind of round, what do they look like? They almost look like the crackers you put in soup, right? Like that kind of a little weird shape. So each of those has an ovule attached to it. So I will highlight the ovule here just so you can see. On the drawing, it's in red. I guess I'll use, oh, green might not be good. Let's use, well, I'll use bright blue. How about that? So here, this is a little ovule peeking out. So in the drawing, they're red, so it's a little easier to see. On this actual specimen, they are also red, but it's a little harder to see because the megasporophylls are also kind of red in color. So that's an example of the megasporophylls arranged in a strobili. And as I said, the leaves don't really look like how you would think a leaf looks like, right? They're modified to be very, very small. It doesn't look really like what you would normally call a leaf. But when the species of cycad has its megasporophylls arranged in a fertile crown, those, or a crown of fertile leaves, fertile crown, same thing, those leaves look more like leaves that you are used to seeing. Pull this up. Oh, pull this up here so in case I want to see where I'm going. Yeah, so those look way more like a leaf that you're used to. Still maybe a little different, but it looks more like a leaf than the leaves that were on that strobilus, right? Oh yeah, here. And you could see, I'm not going to draw them this time, but you could see down here, these red guys, those are all the ovules that are attached to those leaves. So this is what I was kind of getting at, is that likewise, there's a lot of diversity in whether the plant has megaphylls arranged in a strobilus or a fertile crown. Then there's a lot of diversity in what the strobili look like, if it has strobili, depending on the species. And then there's furthermore more diversity on what the actual megasporophylls look like. So, do I highlight it? I do. The megasporophylls that are in a fertile crown look more like a leaf, And you could see those round things are ovules in all of those cases. Yeah, oh yeah, it's on the next slide. Whereas the megasporophylls of Astrobolus have been modified to be very small. They're non-photosynthetic. They don't need to do photosynthesis. They're just holding on to the ovule. And so they've been modified to be very, very small. And the number of ovules per megasporophyll is going to depend on the species. So in the case of the one on the left, it has 1, 2, 3, 4, 5. The one in the middle has 6. The one on the right has 2. And remember, cycads are strictly dioecious, so they have male plants and female plants. And the male plants are going to bear only the male strobili, meaning, male plants only bear strobili. They don't have fertile crown. They always have their microsporophyls arranged in Astrobolus. And the Astrobolus is, as we know already, stem, leaves, and then the Microsporangia will be on the surface of each leaf. And like the ferns, they're on the bottom side of the leaf, right? Abaxial side. So here's what Astrobolus looks like from a male plant. This is one example, right? There's a ton of diversity. There's a real picture on the right. In the middle, you have a diagram. So this whole thing here, it's one Astrobolus, and on it are all of these leaves, right? Oops, hey, where'd it go? One Astrobolus, on it are all of these leaves, the leaves that have Microsporangia on them. So here we took off one leaf. This is one leaf, microsporophyll, and on it are a ton of Microsporangia. This is the leaf in real, so this is one leaf. Next to it is another leaf. We're looking at the bottom side of the leaves, the abaxial side of the leaves, and you can see all the Microsporangia there. All those little dots. Okay. Oh. Well, this will be helpful for you when you're looking at the slides later. So the male Astrobolus there, leaf there, and all the Microsporangia on top. So when we learned about Selaginella and the idea of a microspore and a megaspore, we learned that because the gametophyte plant has to stay inside of the spore, the male and female gametophyte have been reduced, right? Because they have to stay inside of the spore, and the spore is like really small, right? Really, really small. In Selaginella, the male gametophyte plant had been reduced to be a single antheridium, right? So it was only in one organ. That's all that was left, because that's all that could fit inside of the microspore. One antheridium, and that's it. And this is a reminder here. We could see it here, right? So that's the microspore. And then this inside of this microspore is one antheridium, and that's it. That's it. That's all that can fit. So that's from Selaginella. In the Gymnosperms and later in the Angiosperms, the male gametophyte plant has been reduced even further. In fact, both the female and male gametophyte plant have been reduced further. But the female gametophyte plant has gotten a little bit more complex. We'll talk about that in a second. But it's even more reduced. So before we were reduced to an antheridium, and now we're even more reduced. Now we're just reduced to a couple cells. So inside the Microsporangia, which we just saw on the Astrobolus slide, there are those microspore mother cells that are 2N that are going to undergo meiosis to make the tetrad of microspores, right? This should start to be really familiar now. After the Microsporangia, after the microspore mother cells undergo meiosis to make the microspores, those microspores are going to undergo mitosis to make a couple extra cells, and that's what we now call the pollen grain, and that's the highly reduced male gametophyte plant, right? And we call it now the pollen grain. We call it the pollen grain in cycads, in ginkgo, in the conifers, and we call it pollen grain in the angiosperm. So that's what the male gametophyte plant is now, pollen grain. Here is, and so the pollen grain is going to be what vectors the sperm, right? Vector means move, so move the sperm to the egg. And like the plants we already saw in the other labs, the sperms still swim, so motile means that they can swim. But now they don't need to swim through water. They actually swim through, I don't want to get too technical here, through parts of the female plant, and through parts of the pollen grain itself. We'll talk about that. But this is a picture of it. You can see all the little flagella that help it swim. Oh, right here. Yeah, so they swim down a structure called the pollen tube, which the pollen grain will actually grow itself. Cilia, flagella, it just means hair things that move so it can swim. Okay. Pollen grain is kind of a big deal in the terms of evolutionary history of plants, right? Most plants are seed-bearing plants, and most plants bear pollen grains as their male gametophyte plant. So because now we're in the stage where plants have pollen grains, we can actually call the Microsporangia a pollen sac, because the Microsporangia is releasing pollen grains. And that's something I should mention. So these plants shed seeds. So the plants that we looked at before in the previous lab, the ferns, lycopods, and horsetails, they shed spores. Now we're in the seed plants. Seed plants still make spores, but they don't shed them anymore. They shed seeds and they shed pollen grains. So I just want to make it very clear, the microspore doesn't leave the Microsporangia. The microspore, let's go back. The microspore doesn't leave the sporangia because it's in the sporangia. It was made via meiosis, but inside of the Microsporangia, the microspores are going to undergo mitosis and turn into the pollen grain, and the pollen grain is going to be what gets released out of the microspore. So the microspore, released out of the Microsporangium. So the microspore never gets released, turns into a pollen grain, and then gets released. And when we talk about the female gametophyte plant, the seed is what gets released, not the megaspore. But just want to point that out. We're shedding seeds now and pollen grains, we don't shed spores anymore. Yeah, so we can call it a pollen sac. So you can still call it a Microsporangium if you want, but it's also a pollen sac. So it's the same thing because the little Pac-Man's going to release pollen grains. Let's go through this real quick of the alternation of generations in cycads. So as I mentioned, cycads are strictly dioecious, male plants and female plants. Oh, oops. Male plants and female plants. So here we have a female plant on the left. She's diploid, right, because she's a sporophyte plant. So these are male and female sporophyte plants there. It says it down there. And on the female sporophyte plant, in this case of this species, this hypothetical species, their Megasporophylls are arranged in strobilus. So each female strobilus has those Megasporophylls that look like those little things, right? Like those little what are those, oyster crackers? The things that you put in soup. And on each of those leaves, on the bottom side of each of those leaves, there's going to be two, or in the case of this species, 2 ovules, but there's going to be ovules. The number of ovules will depend on species. So those are the ovules. And the ovules are also 2N. Right, and they're on the bottom side of the leaf. Here's our male strobilus from our male plant. And yes, same idea, same idea. There's a bunch of microsporophylls arranged in this structure, and on them there's going to be Microsporangia, aka pollen sacs, on the bottom side. Where do I go with this? Oh yeah, okay. Just so you know what's inside, right? So we're going to look at one ovule here. So this is it, a drawing of it blown up. And inside of the ovule, remember, if I don't mention a term on this slide, you don't need to know it. I'm just going to mention what you do need to know. So inside of each ovule, you have the Megasporangium, right? So I can draw it here. This is the Megasporangium inside of the ovule. Inside of there's going to be the spore mother cells that are going to undergo meiosis. And then ultimately what you're going to end up with is just one megaspore inside. and the megaspore will undergo mitosis and create the female gametophyte plant. A little more complicated than we saw before. On the male Strobilus, we pulled off a microsporophyll leaf. Inside of the microsporangium that's on that leaf are the microspore mother cells that are going to undergo meiosis first, right? Microspore mother cells undergo meiosis so that you have haploid or 1N microspores. Microspores are going to undergo mitosis to make a couple cells and turn into the pollen grain, which is now which is now considered the male gametophyte plant. And that male gametophyte plant, right, the pollen grain, is going to have cells inside of it that allow it to make sperm. Oops, I did not want to annotate. This is a cross section through a ovule. And I think I highlight it in colors. Yes, I do. So that is the location of the female gametophyte plant. Remember, this is there was a Megasporangium which created a megaspore inside, and the megaspore underwent mitosis to make the female gametophyte plant. So that's now inside. And the female gametophyte plant has 2 archegonia on it. We're used to archegonia by now. And each of the archegonia has an egg inside. And so the female gametophyte plant is very well protected, way better protected than it was when it grew outside of the spore, and way better protected than when it grew inside the spore during Selaginella, because there it still kind of poked out a little bit of the spore. Now the female gametophyte plant is super protected because it's not only inside of one layer that you don't need to know the term, and then inside of several other layers too. You don't need to know the term, but I just want to show you how well protected it is. That's what I want you to take away from this slide. Female gametophyte plant and her eggs, super well protected. So eventually the microsporangia or the pollen sacs are going to open, right? Like we saw a little Pac-Man, and release the pollen grains. The ovule, meanwhile, is going to secrete this sticky drop, which we call pollination drop, sticky drop, from the opening of the ovule. And that's going to, so pollen that's floating by on the wind, or pollen that's stuck to an insect, because that's how pollen is vectored or moved in cycads. It's either by the wind or by insects. Let's say it's wind in this case. So wind is blowing pollen grains, and the pollen grains can get trapped on that sticky drop. And that's what you can see here. This circle here is the pollination drop. And then I'll use red just to, and these little dots are, oops, that's not a dot, but the little dots inside, okay, I can't draw a dot. The little dots inside are the pollen grains. When the pollen grain lands on the pollination drop, that is pollination in cycads. That is when pollen reaches that pollination drop, pollination has occurred. So that's a process. Eventually, the pollination drop is going to dry up and go into the ovule via the micropyle. A micropyle is the opening of the ovule. And so pollination drop, which was poking out of the micropyle, is going to dry back up into the ovule. When that happens, it brings the pollen into the ovule. You don't need to know this term, but I'm just saying micropyle will be good to know, but you don't need to know this other term here. Just know that the pollination drop dries up and brings the pollen grains into the ovule, first into one area. And when that happens, the pollen grain is going to grow a tube. And the tube is going to get closer to the eggs. And the sperm is going to swim down the tube to meet the eggs. So don't worry about the terms that I'm not saying here. Just worry about there's a tube that the pollen grain grew. and it made it so that the sperm, which are shown here in squiggly lines, little squigglies, because they swim, are now getting brought closer to the egg, and then they can swim to the egg. And remember, there's 2 archegonia, there's 2 eggs, and there's 2 sperm. Now the pollen grains have the sperm has swam close to the eggs, and boom, the sperm is going to fertilize. And so what happens in the cycads and what happens in all of the gymnosperms, cycads, ginkgo, and conifers, is that even though there's 2 sperm, and even though there's 2 eggs, each of which is in an archegonia, only 1 sperm wins. So one of the 2 sperm will fuse with 1 egg, and when that happens, chemical changes occur. The other egg will be discarded, and the other sperm will be degraded, which is kind of a wasteful process a little bit in the terms of plants. And this gets rectified later in the angiosperms. In the angiosperms, both sperm are going to be used. But in the gymnosperms, only 1 sperm is used, the other one is degrades, and the other egg goes unfertilized. And this is part of the reason why cycads, so it can take months. It can take months between when the pollen grain lands and the pollination drop, right? Pollination. And then it can take months between that event and the event of fertilization in cycads. So that is part of the reason why they're also threatened with extinction in some cases, because that's a really long time to have to wait to have fertilization occur. You know, it's not very efficient. It's not very efficient. Here are what some seeds might look like. Again, there's a ton of diversity in seed shape, size, and color in the cycads. But here's some red seeds, and there will be embryos inside. And those are going to mature on the Megasporophylls. Eventually, the seeds will be fully mature. The embryo will be ready to come out of the seed. Seed's going to fall off the sporophyte plant, female sporophyte plant, land on the ground, germinate, and then grow into the embryo will grow into the new sporophyte plant. And then you're at the beginning again of the alternation of generations. So that is the cycads. Okay. Now let's talk ginkgo. Let's talk ginkgo. So you may have seen ginkgo before. Ginkgo is a very common street tree, as we'll talk about in a couple of slides. This is what the plant looks like. You can see it grows into a really tall tree. This is a close-up of the leaves and of the seeds. So ginkgo, remember, we are still in the gymnosperm, so ginkgo still have naked ovules or later naked seeds. The word ginkgo, so we did ferns last time, right? The word ginkgo comes from maidenhair, meaning maidenhair. And it's because the leaves of ginkgo resemble the maidenhair fern that we saw during the fern lab. Remember, the maidenhair fern is the one that had the false indusium. So the margins of the leaf just bent over to protect the sporangia. That was the maidenhair fern. And the leaves of the ginkgo tree look a lot like the pinnules of the maidenhair fern, and we'll see that on the next slide. And in the phylum Ginkgophyta, I guess I should have said that. Ginkgo is the phylum Ginkgophyta. Ginkgophyta. I don't know if I put a pronunciation on the previous slide. If I didn't, I apologize. In the phylum Ginkgophyta, there is only one genus, Ginkgo. And there's only one species, Ginkgo biloba. Very popular in temple gardens. And even though they're popular in temple gardens and even though they're a popular street tree, we don't currently know if there's any wild population. So all of the ginkgo trees that are known of are ones that people have planted and have cultivated. So there's likely no wild populations left. Here's what I was talking about. So this is the pinnules Remember, pinule is the smallest part of a compound leaf that has many different parts to it, right? Like 1 times, 2 times, 3 times, depending on the compound. The smallest thing is the pinule. So these are pinules of the maidenhair fern. And if you look at the leaves of ginkgo, it looks very similar, right? It looks very similar. So that's why it's called maidenhair tree, because it looks like the leaves of maidenhair fern. Ginkgo is the oldest living seed plant on Earth. On Earth. So you don't need to know the millions of years, but botanists call Ginkgo biloba, the only species in the phylum, a living fossil because it still lives today, and it still looks a lot like it did millions and millions of years ago based on the fossils we find. So here's a fossilized leaf of ginkgo. And this is a not fossilized, I can't even draw. This is a not fossilized leaf of ginkgo. That's horrible drawing. And look how similar they look. They can have slight variations in their lobing, as you can see in the previous slide, right? Some of these have a deeper lobe than others. But the point is that they haven't really changed in hundreds of millions of years. So we call them a living fossil. Very important as a street tree. Ginkgo does not care about living in the city. Ginkgo can handle all the stress that you can throw at it. It's a very, very popular and hardy, like strong street tree, which is great because it allows us to plant these plants in really dense urban sites, and then they can do the ecosystem services that we love that plants do for us, right? They can clean our air, they can help reduce noise, they can provide cooling effects from shade. So very important as a street tree. It's all over here in New York City, all 5 boroughs. Look, this is a whole street of only ginkgo. Tree can get really tall. A tree can get really tall, up to 100 feet tall and up to about 3 to 4 feet wide. So if it can get that tall, but more importantly, that wide, what does that tell us about growth in ginkgo, right? If it can get super wide and super tall, what does that tell us about growth? Hint, it also has like bark and stuff. So it has abundant secondary growth. Look, that's a ginkgo tree. What? So wide, so wide. So abundant secondary growth in ginkgo. Ginkgo is deciduous. Oh, and I should have mentioned, so cycads that we just talked about, they tend to be evergreen. Even though they occur in the tropics, we still consider that evergreen. So they don't lose their leaves every year. Cycads are evergreen. Ginkgo, on the other hand, is deciduous, meaning it loses its leaves every single year, right? So here's ginkgo in the summer, and then here's ginkgo on the right in fall, already has lost all of its leaves. Another cool thing about ginkgo and the fact that it can be planted in so many cities is it has really beautiful golden fall foliage. So when fall comes, ginkgo turns bright yellow or bright gold, and it's very, very stunning. Look, nice. Very relaxing, right? Very, very relaxing. So beautiful golden fall foliage in the fall, which is really nice. Okay, fine. Ginkgo has a shape. So trees have different shapes, right? Trees have different shapes, and we call that their form or their habit. Ginkgo has a shape that looks like, if you're a little kid drawing a tree, here, I'll change color. You'll probably draw something like this, right? Like a circle and a line. Oops, can't draw it, right? Like something like this, if you're a little kid. And it would imply that there's like branches inside, right? This habit of tree, so here it is here. This habit of tree, here's the trunk, and then here's the circle that the little kid would draw, is called you can use either one, decurrent or deliquescent. I like deliquescent because first of all, it's a pretty word. But second of all, deliquescent comes from the Latin deliquescere, which means to dissolve. So it's like the branches come off the trunk and then dissolve. Oh, just dissolve right out, right? The person who used to teach this class, Dr. Casper, he used to say things like, you know, if I'm having a drink on a hot day and I put ice inside, I watch it deliquesce away, you know, things like that. So I prefer deliquescent because I think it's a pretty word. Decurrent is also fine. And these are some other tree shapes. So ginkgo has the one on the right, decurrent or deliquescent. A lot of the conifers that we're going to look at on the next portion of this are going to have what's called excurrent shape. So if you've ever seen a Christmas tree, right? If you've ever seen a Christmas tree before, that has what's called an excurrent shape. So like a triangle on top of a trunk, as versus the decurrent deliquescent, which is kind of a round thing of dissolved branches on a trunk. And then as we saw for the cycads, another type of tree shape is this one on the left, which we can call columnar. Remember, cycads also had a bulbous habit, but other things have a columnar habit, like palm trees, things like that. Okay, or even tree ferns have a columnar habit. This is just telling you more about the leaf, right? So if you've ever come across one of these leaves when you're walking around, you'd be like, oh, this is ginkgo. This is the only species in the family, or in the phylum Ginkgophyta. And so, yeah, this is what the leaf looks like. It has 2 lobes and a really long petiole. That's all that I care that you know about that. Here you go. Oh, yeah. So, yeah, see, it looks a lot like that maidenhair fern pinule, right? You can see why we call Ginkgo biloba the maidenhair tree. Looks a lot like that maidenhair fern pinule. All right, lab mentions this, so I have to mention it too. In ginkgo, you have growth, right? Primary and secondary growth. And when I highlight this for colors to help you. So here, oops, I gotta get out of that. So you have apical buds, which we've already learned about before in the twig lab, right? So we have apical or terminal buds that are going to be at the tip of the branches. And the apical buds are going to produce this long shoots. So remember, primary growth is for lengthening. Apical bud is involved in primary growth. And so it's going to make those really long shoots, which are shown here in blue. The lateral buds, or remember we had axillary buds, but the lateral buds are going to produce what are called the short shoots. And so those here are in red. The short shoots are in red. Long shoots are blue, short shoots are in red. Here's another way to look at it. Oops. Right, long shoot is the thing in the middle. Short shoots are coming off to the side. And notice that the leaves emerge from the short shoots. And the short shoots emerge from the long shoots. The long shoots have nodes and internodes. Does it show it on this? Kinda. Yeah, so like this would be a node. This area here would be a node. And there would presumably be another node down here. And then this would be the internode there. The long shoots have nodes and internodes. The short shoots do not, right? So you can't really discern or tell apart nodes and internodes on the short shoots. It's too short. It's like the stuff is too close together to tell. Long shoots, you can see nodes and internodes. But see the leaves emerging from the short shoots? There you go. Yep. Short shoots emerge from the long shoots, and the leaves emerge from the short shoots. Because ginkgo have this short shoot and long say that 10 times fast. Because ginkgo has long shoots and short shoots, it makes it really easy to identify ginkgo in the summer or in the winter time. So when the leaves fall off and the tree doesn't have leaves anymore, you can still tell it's a ginkgo tree because it has this really kind of peg-like look on it on the branches, right? So it's very noticeable. Okay. Ginkgo is also strictly dioecious, right? Ginkgo also has female plants and male plants. Female plants bear ovules. Male plants bear strobili. So So that's a similarity, right? In the cycads, the male plants always bore strobili. Well, in the ginkgo, the male plants always have strobili as well. And strobili consists of the same thing as they did before, but they look a little different than they did in cycads. Female plants, though, it's a little different than what we saw in cycads. So let's look. So here's some female ovules. And in ginkgo, the ovules are just naked and paired on a stalk. So here is a stalk paired on a stalk. There's one, there's another one. Here's another stalk with another pair. Here's a close-up of a pair. One ovule here, one ovule here, here. Yeah, so the ovules are now, in ginkgo, paired on a stalk. And that stalk, you might notice, is coming out of the short shoots, right? Where the leaves also come out, right? And so these stalks are actually reduced megasporophylls, such that now the stalk is really just a petiole. The petiole of the leaf, there's no leaf anymore except for that petiole, and now we just have two ovules paired on a stalk out and exposed. So this is an example of, we saw this a little bit in the ferns, where you have leaves that are dedicated to photosynthesis and then leaves that are dedicated to reproduction. We call that dimorphism. Di means to, morph means form. So it's kind of leaves with two different forms. You don't need to know the word dimorphism, but just know that, right, there's leaves here now that are dedicated to having the reproductive structures on them, and there's leaves that are dedicated to just doing photosynthesis, right? Okay. Kind of like what we saw in previous plants. This is what I just said, so I don't need to repeat it. You can just read it, but yeah, paired on a stalk, which is a petiole. Here's a close-up of the ovules of ginkgo, so you can see them with the naked eye, right? They're exposed. Like the ovules in the cycads, the ovules in ginkgo also produce a pollination drop. You can see it coming out of the micropyle. Here it is right there. There's another one right here. So that's that sticky drop that allows pollen that's in the wind to get trapped. Same parts that we saw before. Same parts that we saw before, that you don't need to know those terms. I'm just showing you that the ovule is still the eggs are still very well protected. The female Gametophyte plant is still very well protected because ginkgo have ovules. Now let's talk about the male plants. The male plants, like the cycads, have their microsporophylls arranged in strobili, but they kind of almost don't look like leaves. So you can see them here. I think we have a better one on the next slide. But just for location, they also emerge from the short shoot. So here's a strobilus here, here's a strobilus here, and then all the photosynthetic leaves are above that. Here we pulled off one strobilus. It has the same parts as before, in that it has a stem. And then there's these little leaves, which are technically microsporophylls, and each one will have two, here's a pair right here, 2 Microsporangia per little leaf. Here's a close-up, even better. So you can see, here's one Microsporangia, here's another one. Here's the stem. Here's the little leaf. As I mentioned, wind is going to be what brings pollen from the Microsporangia to the ovule. So the Microsporangia, or the pollen sacs, will open and the pollen grains will fly out. And compared to the cycads, the fertilization, so between pollination and fertilization, the amount of time between those events is much faster in ginkgo. Hence, they're, easily cultivated, and another reason why we probably use them as such a prolific street tree. Yep, and so the alternation of generations in ginkgo is basically the same as cycads. So you can go back to that section to have a refresh of what the players are. The only differences The only difference is between Ginkgo and Cycads is now in Ginkgo, the ovules are paired on a stalk, whereas in the Cycads, they were on leaves of a strobilus or on leaves in a fertile crown. Ginkgo only uses wind to move its pollen, whereas Cycads use wind and insects. And then Cycads take a really long time between pollination and fertilization to occur. Ginkgo takes a much shorter time. Those are the only differences in the alternation of generations. Here is some Ginkgo seeds. So remember, the only plants that are going to produce the seeds are going to be the female sporophyte plants because Ginkgo is dioecious. Eventually, the seeds mature. There will be an embryo inside of the seeds. And the embryo is very well protected in Ginkgo because it has a seed coat. Right. It has a seed coat, which we've learned about in the seed lab. But then there's also, the seed coat has many different layers, such that it's really, the embryo is really, really protected. And to be fair, if you were to look at this, you might think that this was from an angiosperm. This kind of, with your eye, looks like a fruit. But just know, morphologically, it's not. It's still technically a naked seed because all the parts that you're seeing here are only the seed. So the fleshy stuff on the outside is part of that seed coat, and the embryo is inside. There's no container that this seed is in. It's still out. I will say, Ginkgo seeds smell. They smell bad. And so, but there are male and female plants, right? And so there's some areas of New York City where, for whatever reason, whoever was doing the planting there planted only female plants for like blocks. And there's every time when the seeds are ready, every time once a year, sometimes it'd be really stinky. It kind of smells like rancid butter or something. It was bad urban planning to do that. Nowadays, most of the time, it's male plants that are planted because they won't ever make the stinky seeds. But maybe you've walked down a block one time and been like, what is that kind of rancid butter smell? Now you can look down and say, oh, look, they planted all the female ginkgo trees here. Okay. I'm going to take let's see. I'm going to see if I can. I'm going to pause real quick. I think we can get through conifers, but I'm just going to take a pic. I realize I can pause, so I'm going to pause really quickly, and then I'll be right back with you to do conifers. I'm going to pause. Okay, so I did take a break, even though it doesn't seem like it to you guys. Let's go. So let's close this out. Yep. Cool. All right, so let's finish out the gymnosperms with the conifers. So we are still, like I said, in the gymnosperms. Conifers are in the phylum Coniferophyta. And here are some examples of conifers. And the reason why conifers are called conifers is because they bear cones. So, or they bear or have cones on them. That's what bear means, bear cones. So the word bearer means to bear or to carry, and conus refers to a cone. So the word conifer means to a plant that bears cones, to bear cones. So only conifers have cones, right? Only conifers have cones. Conifers are large trees, so they have abundant secondary growth typically, abundant wood, abundant bark. And the largest trees that exist in the world now are actually conifers, believe it or not. Believe it or not. Yeah, look at this. Whoa. So just to give you a little perspective here, let me get a different color. Use red. Okay, so on the bottom right of this slide, this is supposed to be a person. Do you see this little dot? That little dot down there? That's supposed to be a person. So let's say around a 6 foot tall person, right? Yeah. So that gives you a little perspective of some of these really, really, really tall trees. Really, really, really tall trees, right? Some of the ones the only one we've come into contact with so far has been the Douglas fir, right? But look how tall that can get. It can reach up to 250 feet tall. That's pretty tall. The tallest living tree in the world is in California, and its name is Hyperion, and it's a type of coastal redwood or sequoia. So you might have heard of somebody talking about sequoias before, or maybe you've gone out to Sequoia National Park, but look at this. So this is a person sitting next to Hyperion, and that's a really massive tree. This picture on the right is real. This is a real picture. It might kind of look not real, but you can Google Hyperion. It is a real tree. It's over 400 feet tall. It's the tallest living tree in the world, and it's a conifer. Here's so this is it's like my New York is showing right here. Yeah, look. So this is Hyperion compared to. All right, so it's almost 400 feet tall. Sorry, it's just shy of 400 feet. And this slide might be a few. I don't know if it says no. This slide might be a few years old now, so maybe it's a little bit taller than this now. Right now, it's in the 380s or so. But look, compared to the Statue of Liberty, it's bigger. It's bigger than the Statue of Liberty. I think that's Big Ben next to that. Really big tree. So the tallest living trees in the world are conifers. The oldest living trees in the world are also conifers. So in California, California's got a lot going on for it plant-wise, is the oldest living trees in the world. They are called the Bristlecone Pines. They are a type of pine tree in the genus Pinus. Pinus is the pine trees. And it's more than 400,000 years old, almost 4,500 years old. Not all parts of the tree are alive, but the parts that are alive are around this old. And the way that they've determined that is doing different types of molecular analysis. People are actually studying these trees to try to understand aging, right? Senescence is the type of science that is, understanding aging, because they're trying to figure out metabolically what's happening that these plants can be alive this many years. So the conifers, right? Conifer Phyta. There's only about 500, 600 species. Most of the species are evergreen. We saw in the first lab an example of conifer that is not evergreen, which was the dawn redwood. So some are deciduous, but most are evergreen. Evergreen just means the leaves stay on year round. Conifers dominate the northern latitudes. What does that mean? So if the farther away you go from the equator, the farther north you go from the equator, at a certain point, the forests are made primarily of conifers. And these forests are what are called the circumboreal forests. Let's look on the next slide. Circumboreal forests. So this is a picture, or it's not a picture, but this is a schematic, or I guess even a map, of the top of the Earth facing the North Pole. So the North Pole is in the center of this. Sorry, the North Pole is in the center of this slide. Here's the North Pole right there. And look at the circumboreal forest. So the word circumboreal means circum means around, and borealis means the north. So these are the forests that circle the north, right? Whoops, whoa. Can't draw to save my life here. Circles, forests that circle the north. Here's the north there. And in those forests, this is the largest forested region in the world, larger than any forest you've heard of before. And it is dominated by conifers. So if you look just at the Russian side of the circumboreal forest, this is about 12 million square kilometers of area. And this part of the circumboreal forest alone is larger than the Amazon forest. And then Canada contains the 30% of the world's circumboreal forests, the rest of which are in Russia. So what does this mean? Well, this is such a large amount of plants that the forests and the lands that are adjacent to those forests contain 5 times as much carbon as the temperate forest and 2 times as much carbon as tropical forest. So remember, during photosynthesis, plants absorb carbon dioxide out of the air and turn that carbon dioxide into sugar. And so if the plant is living, that carbon dioxide gas is being taken in and stored as sugar or carbon in the plants. So that's a really important thing for our atmosphere, is these circumboreal forests. Conifers are important for a lot of different reasons. The one I just mentioned about taking CO2 out of the atmosphere. But we use conifers for a lot of things. We make paper out of them. We build houses with them and floors and tables, and we use them for fuel. And we actually use them for different products. I mentioned in the first lab that junipers are used to make gin that people drink. But turpentine is also something that's derived from conifers. Pine nuts. So if you've ever had pine nuts, they're not really nuts. They're not really nuts because nuts are angiosperms and conifers are gymnosperms. But pine nuts come from pine trees. And then resin and even jewelry in the form of amber jewelry. Actually, I'm ironically wearing amber jewelry right now, so that's a little weird, but I didn't think about that. Yeah, so what are a lot of stuff comes from conifers. So conifers live primarily, they dominate the circumboreal forest, but they live in other places all around the world. They're very common in our area. If you even in just walking around campus, you'll see a ton of conifers. That's why we included them in the campus tour in the first class. But conifers tend to live in very dry areas. So we call them xerophytic plants. So phytic refers to a plant, right? Phyte. And xeros refers to dry. So maybe you've heard of xeriscaping. Sometimes people, instead of planting lawns or whatever, particularly out west, they might just use rocks and things like that because it's so dry. So xerophytic plants live in very dry environments. And think about it. If you're living in the circumboreal forests, there might be a ton of snow and things, but you can't access the water. The water is frozen. So even though it's all around the trees, they can't absorb it because it's frozen. And then a lot of conifers, particularly in New Jersey and the Pine Barrens, live in really sandy environments where there's just not a lot of water. So they're adapted to these dry conditions. Part of the reason they can live in these dry areas is because they have leaves that allow them to hold on to water better and reduce water loss. So conifers have needle-like leaves, which we're probably the most familiar with. Some conifers have scale-like leaves, like juniper has scale-like leaves. Some cedars have scale-like leaves. Some conifers have awl-like leaves. I think we talked about awl, but here's a picture of an awl. An awl is a tool that's used to do leather working. And so these leaves are kind of awl-shaped. And then some conifers do have more laminar leaves. So laminar, think blade and lamina, just means flat and green. So some conifers do have more laminar type leaves. So the main reason that conifers can live in dry environments is because they have sunken stomata. So remember stomatal apparatus, the hole with the guard cells on the outside that regulate the opening and closing of the hole. Here I've drawn circles around all the stomatal apparatuses, that are some of them here, and they're actually sunken into below the epidermis. So that helps them, if wind is blowing on them, the stomata are deeper into the surface of the leaf. It helps them hold on to water vapor better. So that's one way that conifers are able to live in dry environments. So they have this sunken stomata. They also have a thicker cuticle. So if you've ever touched a pine needle, it's like pretty thick. The different shapes, like the needle shapes, scale shapes, allow the leaves to have less surface area, less area for you to lose water. And then they have no spongy mesophyll. So they have a reduction in the mesophyll layer of the leaves. You don't need to know these tissues. Just know that the mesophyll layer is reduced. There's no spongy mesophyll now. Some conifers make resin. It helps them stay healthy. It helps prevent fungal infection. It helps prevent insects from getting inside, and insects and other animals don't really want to eat something that has resin inside of it, so it makes it so that animals don't really want to eat it. This is what I was talking about before, though. So amber is ancient resin that has dried over evolutionary and geological time to be like almost a hard rock. And sometimes if you're lucky, look, I'm wearing amber right here. Wow. Coincidental. Sometimes if you're lucky, you might have small animals trapped inside the amber. So if you've ever seen Jurassic Park or something like that, right? In the very first Jurassic Park, they get the DNA from the dinosaur out of the mosquito that's in the amber, and yeah, okay. But the point is that yeah, sometimes animals are trapped inside. There's actually a colleague of mine at NJIT, New Jersey Institute of Technology, who studies ants in amber. Dr. Phil Barden. Okay. Most conifers have a taproot system. Just a reminder of what a taproot system is, as versus a fibrous root system. Go back to the root lab if you want to learn more about that. This is all stuff we've covered about growth and tissues. So we already covered this, but just know that conifers have abundant secondary growth. This is a slide showing a cross-section through conifer. I think we even saw this slide maybe back in the STEM lab. Just to clarify, some people so in terms of aging a tree, a lot of people didn't get that right on the exam. So if you were to look at this tree, you would say, how old is this tree, right? And the little whoo, I don't know if you heard that noise. If you were to look at this tree, you'd be like, how old is this tree? And on the exam, people put all sorts of crazy answers. But let me just show you again how to tell how old a tree is. Remember, what you want to count is the annual rings. One annual ring is made up of spring and summer wood. So this here is one annual ring. This here is one annual ring. So we can say without a doubt this tree is at least 2 years old. You don't count this part. You don't count any of this stuff. You only count you don't count this. Because that's not an annual ring of secondary xylem. That's the remaining of the pith. So just so you know, if you ever are trying to count how old a tree is, you only count the xylem annual rings. Okay, some of you got that wrong on the exam. We already learned about bark. All right, let's get through this, and we'll finish up with conifer reproduction. So unlike cycads and unlike ginkgo, conifers are usually monoecious, so sex organs on one plant. Just like cycads and just like ginkgo, the male reproductive structures are in a strobili, but now the female reproductive structures are on cones. They're on cones. There you go. Male reproductive structures on the bottom in the strobili, female reproductive structures in a cone. The strobili have on them the Microsporangia, or the pollen sacs, that are going to release the pollen grains. So here is a picture of the strobili on the left. Look at all that pollen coming out on the right. They're going to release it into the wind. Female reproductive structures have ovules and seeds that are on cones. So you can see here each of these little kind of round things are the seeds or ovules. Kind of hard to tell which is which right now. Probably ovules because the cone is not open yet. So remember, in cycads, the way that pollen was vectored or moved was via wind or insects. In the ginkgo, it's only with wind. And in conifers, pollen is only moved by wind as well. So it's the primary method of seed dispersal. The pollen grains it's really cool actually. The pollen grains have these little air bladders that help them stay in the air longer. And not only that, but the air bladders will be in different angles depending on the species and help the pollen grain fly the right way into the cones, because they have to land a certain way in order for them to interact with the ovule. So those are the air bladders, and I always like to say, like when I look at this, I can't not think of something. So here's the air bladders, and maybe you're seeing where I'm going with this, but in my opinion, the pollen grains kind of look like Mickey Mouse. This is what Dr. Casper used to always say too, right? So the pollen grains look like Mickey Mouse. The two air bladders is like the ears. All right, but then when you look at it real, maybe it doesn't really look like Mickey Mouse so much, but come on, this this, sorry, bear with me. Come on. Look at, I mean, come on. Mickey Mouse. Look, here's the eyes. Oh, well. You get the idea. Yeah, so here's what pollen grains look like under the microscope. Similar to what the pollen grain we already talked about before, it's basically a few cells. So the male Gametophyte plant has now just been reduced to a few cells. You don't need to know the name of the cells. So wind is what moves the pollen. Wind is also what moves the seeds. So the seeds of conifers tend to have wings on them, which helps increase their airtime. So here is a seed, and here's its little wing. Right? But as we learned in the first lab, there are some conifers that have slightly different looking cones that are a little more fleshy, and those cones are eaten by birds and bird dispersed. See? So the birds will eat the conifers cones, these fleshy cones, poop the seed out somewhere, and then it'll grow into a new tree. All right, now we do alternation of generations. It's basically the same. It's basically the same as what we saw in Cycads and what we saw in Ginkgo. The only differences now are where the female sex organs are. So this is an overview of it. I don't need to go into this whole thing because we've already gone through alternation of generations several times. But the main difference here is the cone. So basically on the cone, here we go. This is what this is a strobilus. So here's a cross section through a strobilus, and you can see the Microsporangia. Sorry, yeah, the Microsporangia is here's one, full of microspore mother cells, and then ultimately full of pollen grains once the microspores undergo those processes. So there's strobilus of a cone. Sorry, there's a strobilus of a conifer. And then the cone. So this is the only thing we do kind of have to talk about in terms of how alternation of generations is different, because now we have this cone to deal with. So to just keep it organized, in the cycads, the ovules were on leaves that were either in a fertile crown or in a strobilus. In Ginkgo, the ovules were paired on a stalk. Now the ovules are part of a cone. And so basically, I think I highlight the different parts here. Do I? Yes, I do. So basically, you have these scales. You have these scales, and sometimes the lab will say olivorous scales. That's just saying, remember, ferrous means to bear ovules, but scales is fine. So it's scales that have ovules on them. So here is the scale right here. Here is an ovule on that scale. And underneath each scale would have been a little leaf or a bract, which has been lost in a lot of cones. A lot of species have lost this over evolutionary time. Remember when we saw Douglas fir and it had those little leaves? That's an example of a cone that still has the leaves present. Don't worry about all this stuff. It's just, again, the point is that the ovule now is on a scale. The ovule is now on a scale. This is just walking through the alternation of generations. There's a ton of terms here you don't need to know. So again, I don't want to, yeah. Let's just see if there's anything here that I want you guys to know. Not really. I mean, it's the stuff that you should already know, basically. Megaspore, microspore, pollen grain, archegonian, eggs, scale, female gametophyte plant, pollen tube, sperm fuses with the egg, you get a zygote, right? Sorry, the color scheme was not what I have been doing before, but just for time. Okay, so ultimately, pollen grain is going to fertilize the egg inside of the ovule. That's going to result in your 2N zygote, which will grow into an embryo. the seed's going to fly away using its little wing, land, and grow into the new sporophyte plant. Okay. Oh, boy. All right, so we have finished the gymnosperms. We have finished the gymnosperms. So that means you have a homework assignment, right? You have a homework assignment for every class. The homework assignment is available. This class is on Thursday, the 25th. So it'll be available at 8 p.m. starting that time. It is due by Saturday, the 27th. Due by Saturday, the 27th at 6 p.m. The lab exam is 1 week from this class. 1 week from this class. So the study guide is on Blackboard. Make sure you look at it. And then next time, it's our last class. But it's the angiosperms, which is really fun. It's a really fun class. So thank you guys so much, and I will see you next time.